Yep, I think I'm online again. Hi, everyone. Hello, and welcome to another Saturday live stream. I'm Oliver. Let me quickly get again the sound and everything adjusted here. Yeah, here I am again. Today, again, on a Sunday and not a Saturday. A second time in a row already. For private reasons, I could not do the live stream yesterday, and therefore I decided to move it to today. I know it's not the most convenient time for everyone, but still I'm happy that you could join me, and I still hope that it's going to be okay for you. Today I would like to kind of do a little bit of a follow-up of last week's live stream. Last week I put some sand samples under the microscope, and there has been a request to look at those sand samples also using polarized light microscopy. And this is what I would like to do today. This is something that I would like to do today. A little bit of a tongue twister. Okay, so what you see right now over here is the sand under polarized light, and I would like to also explain you a little bit my setup that I have. Of course, it's not the first time that I'm doing polarized light microscopy, but I do want to, but maybe some of the samples that I'm going to show you are going to be a little bit new to you. Well, as always, a big hello all around the world. Usually the first few minutes, I always check from where you are. Yeah, so from Montreal, Canada, from Germany, New York, from Belgium, the UK, Belgium, sound and video, okay, thank you very much. It's always appreciate this feedback. A couple of months ago, all of a sudden, everything was interrupted. No more video, no more sound. And in this case, if really for whatever reason the live stream starts to become interrupted, I will try to restart it, possibly using a different link. So you have to be a little bit patient. It only happened once in the last couple of months. From Germany, from Spain, from Tennessee, the United States, from Denmark. Okay, cool. So glad that you could join me again today. And what I would like to do is I would like to first show you a little bit my setup that I have. Okay, again, polarized light microscopy is very easy to do. So I've made already several live streams and videos using polarized light, but not so much using polarized light microscopy with a stereo microscope. So most of the time I've been using my compound microscope. Most of you might also have compound microscopes. They can be easily converted to polarized light microscopy as well, but I would like to show you how this actually works with a stereo microscope. Okay, so there's also a hello from Sudan and from Illinois. Welcome all around the world. So what you see over here is some sand, and the slide, just if you're new here, by the way, just a few rules of the game. You can, in the chat of this live stream, you can ask any questions that you want. If they're microscopy related, even if they're a little bit off topic, as long as they're microscopy related, I will try to answer them. But please do write an @Oliver or @Microbehunter in front of your question, so it's easier for me to find the questions, because some of the viewers will also try to communicate with each other or using the chat, and then there can be a lot of chat messages. It might be a little bit difficult for me to find the questions that are for me. For this reason, please always write it that you want to ask me something, okay? So first of all, if you were not around last week, you can of course view. It's been recorded, but the sand over here that you see right now on the sand slide that I've made, this one is, where's this from? From Dubai. Yeah, this is sand from Dubai, which I got, about 20 years ago or so. As a present, okay? And I'm now using polarized light, and I'm going to explain you first the setup, because some of you who might be a little bit familiar with polarized light already might realize that something seems to be missing. Something is missing. You can see, of course, this round filter here. I'm going to put the slide away. This is a polarized light, linear polarized light filter. Then over here I put the sand sample. You can rotate, you can turn the sample until the background becomes black, but you might realize that something seems to be missing. And the second thing, it's not missing, but it seems to be missing. That would be the second polarizing filter on top of it. Okay, as a matter of fact, I put it into the microscope itself. So what I'm going to do now is I'm going to remove it from a microscope. Okay, and I'm going to show you what I have here. So what I'm taking it away from my microscope. Okay, yeah, just a second. Just a second. I'm removing something now, and I'm going to show you now my desk view. Okay, and I'm going to quickly show you what I got here. Okay, yeah. So and here this is on the bottom. So what I have is on my microscope. I've got this linear polarized light filter. It's a little bit dirty already. Then you put the specimen on top, and then what you do is you put a second filter over it. Okay, but instead of placing it directly on the slide like this, I decided to put it into the microscope. And what I have here is an adapter ring. Okay, this is for a lamp, and I was very lucky because this filter just fit directly in here. So that's pure coincidence, really. Okay, and then I put it on the microscope, and this makes it a little bit easier for me to do polarized light microscopy because I do not have to put it directly on top of the slide, right? So I have one element less which could be in my way. Yeah, and this is basically what I have done. So if you kind of wonder what this adapter is really for, so I can also show this to you. I will not be using this today, but I have also a ring lamp. Okay, with light coming from the top, and then this actually goes on the bottom of the microscope, and this kind of goes in here, yeah, and the screws kind of hold tight. Yeah, so that's the reason why I have this, but I do not need this today, so I'm going to put it away, and I'm going to use this here now, yeah, as a filter. Okay, so this here is these are glass filters. However, if you do not have those glass filters, it is also, here it is, it's also possible to buy yourself those polarized foils. This is not protective plastic on top. They're fairly cheap, and they can also be used, and then you can actually use scissors and cut out whatever you want. Okay, so but I will not be using this here because I've got those filters, but it's actually exactly the same. Yeah? Please make sure that you get linear polarizing filters. They're those 3D paper glasses, and they are circular polarizing, and they don't quite work as well. Okay, so what I'm going to do now is I'm going to put this back again on my microscope. Okay, yeah, and so you won't be able to see that now, but yeah, so basically you see here it is in the bottom, yeah, and I'm going to put it all the way up now into onto the objective. Okay, and now I can do again some polarized light microscopy. So what I would like to also explain now a little bit is the problem that I'm having here, and that is it's not a big problem, but I've got a new HDMI camera. Generally, I'm very satisfied with it. But what this camera does is it also automatically adjusts its exposure time. And sometimes if you just, now it's kind of dark, and then sometimes it will actually become brighter again because it adjusts exposure time, okay? So it just wants to make you aware that this is an issue, could be an issue here, okay? So here we go again, yeah, here, this is again the sand sample, yeah? So this is basically the setup that I'll be using. And if I want to make the background dark, all I do is rotate this filter, and then you can actually see that, yeah, now it's brighter, and then when I rotate it, it becomes a little bit darker, and it's actually the nice way, okay? So I'm going to interrupt myself again. I'm going to quickly check, okay? From Denmark, from Illinois. Would you put some dog saliva under the microscope? Well, I do not have dog saliva, unfortunately, right now, but I have dog hair, dog fur. So that's actually a dog fur that I also prepared. So this is the fur of a dog. Yeah, and look, this is how it looks like under polarized light. Yeah, it's a little bit overexposed. I will also explain why this is the case. Yeah. So if I understand, so that's a question for me now. I hope I'm able to answer it. So if I understand correctly, the same setup can be done with a compound microscope by sandwiching the slide between the polarizing filters. Absolutely correct. Okay, absolutely correct. If you want to, I can actually also show this to you using my compound microscope. However, that's also important. This with a compound microscope, I only would use the low power 4x objective because then you still have enough distance. Yeah. As a matter of fact, I think that it might even work better with a compound microscope sometimes. Let me just quickly get my compound microscope also set up so that I can actually also show it to you. Okay, so what I'm going to do now is I'm going to show this quickly to you, how this works with a compound microscope. Okay, you see it's kind of funny. I'm not seeing any image now for whatever reason. Uh-huh. This is a little bit disturbing, I have to tell you, because I do not get a picture with my compound microscope for whatever reason, and I don't know why. You see, everything's kind of dark, and this is a little bit, how do you say, not so cool. Ha! Yeah, it works. I'm so happy. Here we go. Okay. Sometimes there is a USB connection problem, but I just wanted to show it to you now. How this works with a compound microscope, so that you actually see that the principle is exactly the same. Okay. And now you can look at the top here. And what I recommend is that you do not put it, of course I could try to put it directly over the lamp, but then the polarized light has to go through the condenser, there are optics in here, and so sometimes it actually works better if you just put it directly on here, okay, yeah. Then you put the slide on top, like this, okay, put this away, yeah. Put the polarized, so this is now basically, yeah, of course you have some problems now moving it, and then you put the second polarizing filter on top, and you have to rotate this, okay. I have to remove this other thing here from the top, okay. Okay, and for whatever reason it's too dark. Where is this? Here we go. Okay. Yeah. You see that, yeah, there are those things that are actually changing the polarization of the, some of the sand grains which are changing the polarization will now start to appear to light up, yeah, in such a way. Okay, so it is just it's a little difficult now, the whole setup for me here, but I just wanted to illustrate this principle that this also works. It's kind of dark, I think, but I think this depends quite a bit on the sample that you're using. Okay, so but let me remove this again. Okay, and let me remove this again and put everything back again here. And let's put it again over here, the stereo view, which is over here. And I have to put back again the polarizing filter into the microscope. Okay. And then I can essentially put the other filter again on the bottom, and by turning it, yeah, I'm able to make sure that the background is dark. And what you see over here, this is all dust and dirt. Okay. So, so hello from Texas, from New York, okay. Do you plan to write a book about microscopy? I'll buy it. Thank you very much for the suggestion. I'll be honest with you, I already started to write a book, and I started to write it already many times. I've got a lot of pages together already, but never could really motivate myself to actually get it completed. I lost myself in a lot of details as well. And then I kind of discovered that there are, I still have not found the focus of the book yet, okay? Whether I should make it more introductory or more advanced, or if I should also include a lot of theory or not. So this is a little bit the thing. Thank you for the motivation again, for actually suggesting it again. And I'm going to keep up with that idea, okay? So, so this is something what I would like to do now is I would like to show you also a couple of other specimens before I show you some other sand. So and also some specimens that work quite well and others that do not work quite well, and some of the problems or issues that you have to take into consideration. Okay. So first of all, this one here is the cross section of a stem, and it looks fairly nice. However, it does not quite look as nice as if you were to look through the microscope directly. And that has to do with the fact that many cameras will start, because the overall image has a dark background, it will calculate the average exposure time that's needed. And this basically means that the bright parts of the specimen sometimes appear to be overexposed. Okay, so this is a little bit of a problem. So I just want to show you as well what you have to do sometimes and I don't know if you're able to see this. Yeah, Yeah, you have to sometimes adjust the brightness as well. Okay, and these are now the default values. And I'll just stay with the default values, but I just wanted to clarify this, that sometimes because of the automatic exposure, it's not always, some of the parts seem to be a little bit overexposed. In OBS Studio, you can set the manual exposure in some cameras. That is correct. And this is actually also something that I've tried now a little bit, but my camera, because it is actually an HDMI video camera, will adjust the exposure automatically, but I can determine whether it should generally increase it or decrease it, but I'm not able to set it at a fixed exposure time. Okay, so just wanted to clarify this with you. Look, another thing that you can do here, of course, is with the stereo microscope, you can get more or less continuous zoom. Yeah, and what do we see over here? All of those beautiful tiny little lines are, of course, dust. Okay, so you see that some of the dust is actually changing the polarization of the light, or is depolarizing it, and therefore it will appear bright. And again, what you see, the specimen itself is now way overexposed. So this is something just that you have to be kind of aware of when you're working with images of large contrast, that it's difficult to both get the specimen and the specimen correctly in the proper exposure time. So this is actually one of the specimens which still worked quite well. Okay, I do also have a few others where you do not see the structure quite well, even though these are also cross sections. I think it's kind of fascinating. So this one is a fern. Just want to show this to you as well. Okay, and when you put this one here under the microscope, you're actually able to see that not some parts appear to be bright, but others not. So it's quite interesting, I think, to see how different parts, or different parts of a specimen actually respond different, to polarized light. Since mitochondria in the eukaryotic cell has its own DNA, is it the regular DNA? Are they all identical for a person, or are they identical for a species? That's an interesting one. It goes directly into the heart of biology. It's also called the mitochondrial DNA. First of all, the mitochondrial DNA, and even if it's not microscopy related, it doesn't matter. I'm going to still answer it. Mitochondria have their own DNA. And the interesting thing is that this DNA is inherited over the mother. Okay, so this basically means that the egg cell has a mitochondria, and this is the mitochondrial DNA that we inherit. So all of our cells have, for this reason, the same mitochondrial DNA. However, this mitochondrial DNA is not the same within a species. This is the reason that if you, for example, want to do some genealogy, and if you want to get some, if you want to figure out who your relatives are, or from where you are from, what you can do is you can do some mitochondrial DNA analysis, and then the result will actually tell you from which region of the world your mother is from. Okay, so you can actually go back into time and trace your ancestors, so to say. So simply, so to answer your question, are they identical for a person? So within my person, within my body, the mitochondrial DNA is the same between the cell, but not among different people. Okay. Or are they identical from a species? So within the species, no. So my mitochondrial DNA is different than yours because we've had different mothers. Okay, so just a very quick, So this one over here is another slide, which was kind of surprising, and because what you see over here, those little dots, are actually starch grains, and those starch grains should actually really light up in polarized light, but they don't in this case. Look, and I'm rotating the filter, and it doesn't seem to make a huge difference. But look over here on the side, you see, now the background is black. But over here, this seems to be embedded in some kind of a matrix or in some kind of a medium. And this completely destroys the effect of the polarized light. So you see, yeah, here we're now on the side here, and those starch grains, those little dots here, which normally are really nicely polarizing. Yeah, you don't see this at all right now. Yeah, so it almost looks like, yeah, it's non-polarized. Yeah. So this is also quite fascinating, which I did not expect, because starch grains usually, yeah, they really shine up in polarized light, but in this case not, because this has to do something with the way that the specimen, the slide, was prepared. Yeah. Okay, hello from Athens in Greece. Okay. Okay. Yeah. Okay. Have you ever looked at Romanesque? I have to be honest with you. I don't know what Romanesco is. Okay. Yeah. Okay. So that is a little bit the thing. Okay. Yeah. You could. Oh. Okay. So no way. So interesting. That, I couldn't find the answer anywhere. I think this refers back to my mitochondrial DNA. Yeah. So this, so there's a lot, by the way, concerning mitochondrial DNA. Very fascinating. The mitochondrial DNA is relatively small, and contains mostly genes that are responsible for yeah, cell respiration. So which basically the mitochondria does. So that's another one over here. Yeah, yeah. So this again is stem cross section. And also quite nice. But I know we're going to look at the sand samples in a minute, okay? What software do you recommend for USB industrial cameras? Okay, I'll be honest with you. I'm using, for what I have is the USB industrial cameras. Depends what they, yeah, if they have so-called UVC, I think it's called. It's a general standard. And what you do is they should be able, ideally, you should be able to directly view the camera in Windows. And the software that I'm using right now is OBS Studio, which is extremely flexible. It is a live streaming, like yeah, like what we're doing right now. So you can combine several cameras into one picture, but that's only one possible application. Yeah? OBS Studio, you just can have use it for regular viewing. You can record with it. You can overlay images. You can do get snapshots, screenshots, whatever you want. Yeah. So OBS Studio, which is a free program, once set up, gives you a lot of flexibility. Yeah, so yeah, what I have done is I've got an HDMI industrial camera, and this HDMI signal I'm converting to a USB signal by using a small converter. Okay. Yeah. So yeah. So essentially, those many industrial cameras will work with yeah, like a webcam, so to say. So you can essentially also use the built-in Windows camera viewing software if this is what you want. The reason why I chose an HDMI industrial camera is yeah, simply because I do not need a very high resolution, but I need a very quick frame rate. Yeah? So I need a very smooth image, even if the resolution is not so high. Yeah? Okay, so yeah, so this is again a cross section, yeah. But there are plenty of I'm quite sure there are many other how do you say, types of software out there. Okay, so this is an and this is the fern, and what is this one over here? Yeah, here, and look, when I rotate this here, it's because this is another thing I just wanted to show you. I see some dust and dirt, but you see, it doesn't really light up. Okay, yeah, here it doesn't work. You see over here the background changes. Yeah, the camera does compensate, yeah, for the darkness, but over here again the specimen itself, because of the medium and how it was embedded, it really does not respond to the polarized light so much. Yeah. So again, what we see over here is whether it looks nice or not does not only depend on the specimen, but also how the specimen was prepared. Yeah. Yeah, so this is Romanesco is a fascinating vegetable having amazing fractal patterns. That's thank you for the info. I didn't know that. I think it's the same family as broccoli and cauliflower. You really Google it. Okay, I will try to figure this out, because if it looks nice, then indeed I'm always looking for interesting specimens for videos. Ah, it's a form of broccoli. Interesting. Okay, that's cool. Yeah. So, yeah, so I just wanted to show you again with some plant cross sections, but there's something else I would like to show you. Let's put this away here. Of course, when we're dealing with polarized light, there is no way around vitamin C crystals, which I just made before. A couple of, yeah. So this here is, and again, it's a classic. What I've got here is on this slide, I've got some vitamin C. Okay, see, I'm rotating this now, the filter, until the background becomes very dark. Yeah, and then what you get is you get those nice, beautiful patterns. Yeah, normally I always viewed it with a compound microscope, but I just wanted to show you that a stereo microscope also works. Okay. How did I make that? Again, I made plenty of videos already before. You take a little bit of pure vitamin C, dissolve it in water, put it on a microscope slide, and then I used a coffee heater. Just to show it to you. Something like this, right? It's a coffee cup warmer, and I use that to evaporate the water of the slide, and then crystals will start to form on the microscope slide. I can actually show it to you also using the desk view. Okay, here, this is how it looks like. Yeah, you see the white. Yeah, that's basically the vitamin C, which formed crystals here, and in polarized light, this looks quite pretty. Okay. Yeah, so you put it again. I put it again here on the scope. No, that's the wrong microscope. Here we go. Yeah. I made another one over here as well. You have to play around with the amount because sometimes if you use too little or too much of this, of the vitamin C, then you might not get beautiful crystals. Again, you have to rotate the filter until the background becomes very dark, and then you see over here the different beautiful patterns. Yeah. And those black dots that you see, these are basically vitamin C crystals that have not fully dissolved yet. So, question, did you crystallize yourself the vitamin C? Yes, I did. And what you do is the following. You take some vitamin C in powder form. I've got some, I'll just show it to you in case you have not done this yet. So I took some vitamin C in powder form. Just a second, I'll show you how it looks like. So here we go. Okay, so this is, we're not talking about vitamin C tablets, okay? We're just talking pure vitamin C. Yeah, which is looks almost like salt or sugar. You take a little bit of that and you dissolve it in some water. You take a pipette and yeah, I can actually demonstrate this to you if you want to. But I mean, there's not enough time really to dry it. But what you do is the following. But we could try this as well. Look, you take a little bit of this vitamin C, you put it on directly on here, and then you allow it to dry. So I spread it apart. But what we could try is we could actually have a look how this looks like under the microscope. Wrong microscope again. I think this might be the droplets. See, that's the, yes, that's the, how do you say, let's do it like this. So that's the drop of vitamin C that I put on here. You see that it's dissolving now the crystals, and then if we wait a little bit, we might actually be able to see that it starts to recrystallize. Actually, I wanted to show you the sand. I'll do that later, okay? But yeah. And the evaporation of the water takes quite a bit of time, really, and for this reason I use the hot plate. But what we can do here is we can simply wait for this to spread apart, and then yeah, it will evaporate hopefully, and then it will start to recrystallize. Yeah. So yeah. What I could do, however, also is I could simply take a tip of a pipette and spread it a little bit more. See? Ah, look, the tip of the pipette being made of plastic also changes the colors. Yeah, it's polarizing as well. See? We're now dissolving. Yeah. See, it doesn't dissolve anymore because it seems to be already a saturated solution. Yeah. Yeah, so these are basically, yep, that's a saturated vitamin C solution because the crystals are still visible. Okay. And then now you have to be patient. You have to wait for the water to evaporate. And then the crystals will start to reform. Yeah. So I'll just, so just in case you wonder how this works. Yeah. My dog licked a microscope slide and it did a nice fern crystal pattern on it. I'll try it again with polarized light. Yes. What if you do that? You'll be surprised how, if you take tears, for example, and put it on a microscope slide, it will also make beautiful crystals. I think what you see there is crystallized salt. Okay, so in the saliva and also in tears, there is lots of salt. And when it crystallizes, then of course, when the water evaporates, then of course it also makes crystals. Yeah, so I think it might be salt or other dissolved substances. Yeah. Wouldn't larger crystals form if the evaporation is slower? It could happen. However, this depends also a lot on the crystallization seeds. So sometimes if there are a lot of impurities in there, then it can start to crystallize in many different places at the same time. And then the crystals grow together. And once they've grown together, they cannot become bigger anymore. So I think it does depend on the speed of crystallization. Sure, that's one thing, but also at how many places the crystals start to form. Yeah. Have you tried aspirin crystals? Yes. Yes, I have tried it. As a matter of fact, I've got some aspirin over there. You have to take it in, you have to dissolve it in a little bit of alcohol. Okay, so that is, So the colors of the plastic pipette is stress forces. So I'm going to, as a matter of fact, I did prepare also something else. So I'm going to wait here. This might actually take a little bit of time to dry. But the plastic, I've got a ruler over here. Okay, and look what happens if I put the ruler under here. Yeah, isn't this great? Let's zoom out a little bit. So that is the ruler, and look at the colors here. And these are indeed because of the stresses in the plastic. So if you actually, I don't know if you can see it so much, if you were to bend it, then you can actually see that the color patterns would change. Yeah, look, if I'm pressing it now, so this is actually something that can be seen quite often in plastic. Yeah, that there are essentially tensions in there, and this causes those color patterns. Yeah, so this is quite something quite nice, and that's also the thing that we've just seen before with the pipette here. It's a little bit dusty. Yeah, here we go. Yeah. And I don't know if sometimes maybe if you were to bend it or so, then you might actually also be able to see that the colors might change a little bit. You see, it's difficult. It becomes darker and then it becomes brighter again. And the reason is because the camera tries to adjust its exposure time. Yeah. Yeah. So, yeah, all these strings here, that's all dust. Yeah. So, yeah, but again, here we've got the ruler. Yeah. So let me see here a little bit. This, I don't know if this is. Look, they're already. It's ready. Look, it's starting. It's starting. Here we go. Here. Okay, it's now starting to form to recrystallize. And look how those two circles are slowly starting to grow together. Where is the arrow? Okay, look at this here. Yeah, they're now going to start to grow. And this is actually what I tried to explain before. If there are lots of places where the crystals start to form at the same time, then they won't become quite as large because they will start touching each other. Yeah, so this is, so that's a little bit just something I wanted to, Let's go out a little bit here. I think generally, I think using stereo microscopes for observing crystals and sand and so on is easier than with a compound microscope, simply because you have more space to manipulate the specimen. Yeah. It's very often you can see that the crystals start to form somehow, at the edges, Where's the arrow? Here's the arrow. Yeah, very often you see them starting to form at the edges first. Yeah. So that's actually quite quite a nice thing. That's a lot of music in vitamin C. Yes. What I would like to say is that vitamin C is actually one of the easiest. Not easiest. Is a fairly easy substance to deal with, and generally because it looks so nice and beautiful. Yeah, you can make a lot of nice pictures. Yeah. Yeah, and you have to adjust the concentration of the vitamin C in such a way. If there's too much vitamin C, it's not quite as colorful. Yeah, but you get the idea. Yeah, Sometimes where there's dust and dirt, this will sometimes also act like a crystallization seed. Yeah. Can you rotate the polarized filter to see the cross rotate? Yeah. So basically, I will explain this. Let's look at one of these things. Do you actually see that it looks like that there's a cross inside the filter? Okay. And what I do is if I rotate the bottom filter, then the background will become brighter. So what I have to do is I have to rotate both filters, the top one and the bottom one at the same time. Okay, which is difficult. Yeah. Okay, I'm rotating the top one now alone. Look, and then you can actually see that the cross, in the crystal that they're rotating almost like in a wheel. Okay. Looks quite nice as well. Let's go here. Let's refocus. Yeah. So that's essentially the thing. And of course, what you probably would like to do is maybe some kind of a system where you're able to rotate both filters at the same time. Again, here the exposure is not quite as nice because the crystals are overexposed. The camera is doing that. Yeah. So this is kind of the idea. I also have to refocus a little bit. Okay, so that's basically just wanted to show you a little bit the thing here. Let's move on. Let's move on a little bit. What time is it? Yeah, 37 minutes. I want to show you again a couple of the how do you call this? The sand slides. And I just want to show you that not all of them are really super ideal, but that's the whole point. A little bit. So this is the one that I showed you before, right? And if you now zoom in, then you're able to see that there are some sand grains. Well, let's have a look. Yeah, let's just show it to you here. Let's zoom in. So the majority of sand grains, basically, they look, they have basically the normal color. Yeah. But look at this. Occasionally you're able to find sand grains that actually do have a rainbow color. And what I think happens is the following: those sand grains, they simply depolarize the light. So basically, the polarized light from the bottom filter goes into the sand grain. The polarization is completely destroyed, and then it will appear just like in a regular color. However, in other cases, if there is some kind of a crystalline structure in here, what will happen is that some of the polarized light is not just is rotated and turned, and then this causes those rainbow colors. Yeah. So I think what we have here is cases where we're able to then distinguish a little bit those sand grains that are actually like the ones over here, right? Which are kind of optically active, and others that are just basically destroying the polarization. Yeah, and I think this is one of the reasons why some of them have actually rainbow colors while others do not have that. Yeah. So just wanted to show this to you. So this one again is the sand from Dubai. Yeah, very nice. We have to make sure that the background is nice and dark. Yeah. Yeah. So, and I also want to show you a few others. This one here is Crescent City, Pacific Ocean. Yeah, let's go. Yeah. Because some people emailed me the other day and they were kind of wondering, is it possible to do something like dark field microscopy using stereo microscopes? Basically, where the specimen is bright on a dark background. And it says, well, what you have to make sure is you have to make sure that the background becomes dark. And what we have here now is a case where we can make the background dark simply by using crossed polarized filters. Yeah? And also over here, we see that we do not see so many rainbow colors really, so I think that this is more a case of depolarization. Yeah. And there's a comment, you can sometimes determine the type of stone from the colors. Yes. What you normally, what I'm doing right now is just a little bit of fun, how do you say, fun polarized light microscopy. But if you are actually doing this for research, with a compound microscope, what you actually have is you have a controlled way of turning those filters, and there are actually also degrees on it, yeah. And because you have to actually measure out certain polarization angles and so on. Yeah, of course, I'm not doing that. I'm just doing some basic nature observation this way. Huh? Yeah. So this is Crescent City. What else do I have here? Another sand sample. Yeah, this is from the Netherlands. Would you collect 2001, so over 20 years ago. Yeah. And here again, we're able to see that some of the, here it is actually quite nice, some sand grains actually do show those rainbow colors as well. I think it looks pretty nice. Yeah. So if you really want to do this, I've never done this before, but some folks out there, who are observing minerals, what they're doing is they're actually polishing the minerals in a very thinly and finely, into a very thin cross section. You can do that. It's possible to put rocks also on the compound microscope by really polishing it. Here we've got more of them that actually have this crystalline structure, and which display those nice rainbow colors. Bifringence, yes. Yeah. Have you looked at the diamond particles embedded in various tools such as the saw blade? No, but that's actually interesting. That would be interesting, yeah. To look at, there are, of course, for in saw blades or for abrasive, maybe some substances and so on. There are artificial diamonds in there, which make it very hard. So that and that would be a possibility as well. Thank you for the recommendation. I've not tried that. Yeah, a lot of here, a lot of rainbow colors. And dust, of course. Yeah. Let's have another look here. Kalahari Desert. Yeah, very red. Yep, here we go. Okay. What metallurgical compound microscope have in particular? There is one of the two polarized filters, the objective. What metallurgical compound microscopes have in particular? There is one of the two polarized filters, the objective. Well, let's put it this way. Well, let's put it this way. Metallurgical microscopes actually have light coming from the top. Okay? So that's why I'm thinking a little bit of what this could mean. Because when light is coming from the top, actually in polarized light microscopy, the light is coming from the bottom. You have essentially polarized light from the bottom, then you have the specimen, and then you have another polarizing filter called the analyzer. And sometimes this analyzer is actually inside the microscope itself, not directly on top of the specimen. Yeah? So a little bit from this. So this system does not really, to my understanding, fit together with so-called metallurgical compound microscopes, which actually have the light coming from the top. Yes? So I'm not quite sure how this would work. Okay. And very often, I can actually show it to you in my case. Let me see. I'm going to now go to the scope and just look at the image at the top, right? So I'm going to, because what I have over here, and this is not a what you have is in here. If you can take this out, and this is also a place where you can put in polarization filters and so on. Yeah. In this case, it's not a polarization filter; it's a Wollaston prism. Yeah. But many microscopes, what they will have is they will have these the possibility to insert filters and so on above the objective. Yeah? One of the problems, however, when using polarized light is that if you actually pass the light first through the objective and then have a filter, then sometimes you're losing the polarization because this filter, the objective sometimes depolarizes the light. So what you need is special objectives that are so-called strain-free objectives, which do not change the polarization of the light. Yeah? And these objectives are a little bit more expensive. Yeah? So just some technical side information as well. Yeah? So, you can buy loose industrial diamonds quite cheaply. Some buy them to make their own stone polishing tools. Oh, that is interesting. I didn't know that. Okay, thank you for the recommendation. So we're talking about industrial diamonds, which basically, might also look quite interesting under the microscope. Yeah? I would love to see soil sample next time, nematodes and fungus, especially from different types of soil. Okay, that's an interesting one. I would like to talk a little bit about this because you're asking. Several years ago, not many years ago, but several years ago, I did put soil samples under the microscope as well. So I don't even know, it must have been 2, 3, 4 years ago maybe. And the issue was the following, that essentially it was not super exciting because while I could see a few things, there were not so many microbes, not because there are no microbes in soil, but the density simply was not high enough, and many of them kind of disappeared. They disappeared between the soil particles. Yeah, so it was kind of difficult to see the microorganisms between the soil particles. Yeah, so I saw a lot of amoeba shells and so on, but not so much movement going on. So I just wanted to say that it was not very, how shall I say, very exciting. Okay, because I'd have to concentrate them first to see more of them. So even though they're kind of difficult to see in contrast to the soil particles. But I will try. So what I've done is I've made an enrichment culture and multiplied the soil microorganisms a little bit, and then it worked out quite well and had sufficiently high density for it to be interesting. But I will remember this and think about something. Okay. Yeah. Refractometer. You can have a free down gold dust from your local jewelry. Just ask them for dust sample. Oh, that's interesting. I can imagine because maybe they're cutting diamonds, because I know that sometimes diamonds are also being cut and polished and so on. So maybe there are some, some leftovers. Okay, but thank you for the recommendation. I think it's it's something I didn't consider. Yeah? So, over here, how's this one look like? This one again. Yeah. 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 I do think it kind of does look similar. I don't know. Yeah. So this is from Roscoff. This one is Sunset Beach in Cape Town, South Africa. Look. But maybe one of the differences that you do see is the shape of the sand grains, because some of them are much rounder. So it actually shows that erosion has kind of polished them and rounded them off. Yeah? While others are a little bit more, ragged and have more corners. Yeah? So. But the whole thing with the diamond actually is fascinating. I didn't think about actually putting diamond dust also under the microscope. And what else do we have here? This is Sahara. This is I forgot to put no, I do have a cover glass. This is a sample that was sent to me recently. Because it doesn't look quite as much like sand because it's so small. But here I'm in Central Europe, and a couple of times every year, what we have is when the wind and weather conditions are right, we're getting a lot of Sahara dust, blown into Europe. So the sky does look a little bit reddish, and then sometimes you're actually able to see some of the dust collect, on cars, on the window and so on. And this sample here was sent to me, by a microscopy friend in Germany. And this is basically Sahara dust. And when you put it under the microscope, of course, you see that it looks very fine. Of course, it has to be carried. Wind and rain are able to carry it, so it's very fine. And because larger sand grains cannot be transported, obviously. Yeah. Yeah. Gold dust. That's also something. Yeah. I'm going. Yeah. Gold dust under the microscope. It would be something as well. Huh? Yeah. So this is also quite nice. It doesn't look particularly exciting right now, but considering the fact that this dust traveled thousands of kilometers, from the Sahara, because there was a sandstorm there, and then it was spread across Europe. Yeah, so in that sense, I kind of like the idea. Yeah. So, and what else do we have here? What is this here? Mojave Desert, great sand dunes. So also, Look, this looks nice. Here, very nicely visible that some of the sand grains are bifringent. Yeah, I read you can cut the backlight of a small LCD screen mounted on the top of the condenser and control it with a Raspberry Pi to make programmable filters. How does this work? You can cut the backlight of a small LCD screen mounted on the top of. Oh, I get it. Ah, okay. Because it is possible to make so-called Reinberg filters, where basically you have different colorful discs, and instead of using Reinberg filters, why not directly mount an LCD? That's an interesting idea. Okay, okay, yeah. Nothing to see with gold, I guess. Only well, the atoms, unfortunately, you will also not be able to see because they are way too small. Yeah, yeah. Diamond. Okay, yeah. So gold and diamond dust. If you ask politely, the jewelers might give them away for free. Look at the colors here. It's really nice. Yeah. So So enough of plant cross sections and sand grains. And a little bit of vitamin C as well. Let me quickly put this back into the box here. Yeah, maybe you already know. I've talked about this already. We've got a laser cutter at home. And I've made this box myself using a laser cutter. Yeah. So that's why I also added my logo over here. One of the reasons why I added the logo is so that you do not accidentally open the box the wrong way around. Yeah. So, but that is basically one thing. And I want to try now something else. I would like to show you, unfortunately, something which I was kind of hoping that it's going to work nicely, but it didn't. But it kind of works half. And that is, look, I've got some water fleas here. Where are they? Here they are. And this is actually one of the examples where I was a little bit unhappy because the camera was always adjusting its exposure time, so I was not able to get it completely dark in the background. But the water fleas now also appear to be bright on a darker background, and I have to tell you, it looks much better when you look directly through it than here. Yeah, on camera. Huh? Okay, so this is these are Daphnia and so on, they too sometimes look quite nice if you have them, yeah, in different lighting conditions. However, this is one of the examples where I was struggling a little bit getting a good image quality, as you can see right now, right? Sometimes the background becomes darker, yeah, and then the camera immediately adjusts the exposure time again, and then they become overexposed, yeah. But again, this is a little bit the reason why I'm showing this to you is because I've repeatedly received requests on how can I get a dark field-like image using a stereo microscope. Yeah, and one way that I suggest, so basically dark field light means a bright specimen on a dark background. And one of the things that I've recommended is, I'm going to show this to you, yeah, is to use a, how do you call this, a mirror on the bottom, yeah, with light coming from the top, yeah. So this is now light from the top with a mirror on the bottom. But the problem with that, as you can already see, is that yeah, it doesn't look nice. And one of the reasons why it doesn't look so nice is because you have a double image, because the mirror, the glass of the mirror also reflects. Yeah, so you see, yeah, so that would be a possibility of that kind of getting a bright image on a dark background, but the quality isn't quite as good. Yeah, and the other thing that I tried is using simply a dark, yeah, background, yeah. So that's basically black plastic, as you can see on the top, right? But you also don't get quite the same effect. Yeah, and you see that the black plastic also reflects some of the light. Yeah. So for this reason, I've been yeah, using polarized light, and as you can see, yeah, adjustment is a bit difficult, also because the camera keeps on adjusting the exposure time. Yeah, but it looks better if you observe directly. Yeah. What is the difference between front-mounted and bottom-mounted condensers? Can they be compatible? Front-mounted and bottom-mounted condensers. I am completely at a loss now. Front-mounted. I mean, bottom condensers are normally mounted on the bottom, right? But what do you mean with a front-mounted condenser? I'm a little bit at a loss because yeah, I'm not. Yeah, maybe there's some certain microscope setups where this is the case, but normally condensers are on the bottom. But what do you mean with front? I have to do a little research. I have to say sorry, I don't know. Okay, we need a real matte dark paper. The black plastic is too reflective. That is correct. And what I've been thinking about is this is way too reflective, and I've tried to also use black paper, but there is this paint apparently that some people are using, which gives you a very, very, I don't know how it's called, which gives you a very dark color. So there's this especially highly absorbent black paint, and I don't know where, if it's possible to buy that, but I know that some people actually even painted their cars with that. Yeah. Yeah, and it's totally black. Yeah, so it's the blackest black that you can get, and that's actually something that you would probably need, because if you, yeah, are using this here, as you can see, it's way too reflective. Yeah, so that's a little bit the problem here. Yeah, so yeah, so Vanta black. Thank you. It's called Vanta black or Vanta black, and I don't know if it's possible. I haven't tried it yet, but I don't know if it's possible to actually buy the paint. But that would be, even if it doesn't, even if it just were some kind of a sheet of foil with this on it, would be kind of sufficient. Yeah. Yeah. So that's something that yeah, maybe that's the next next experiment that I'm going to try. So. Yep. Okay. So that's. Is there anything else I can show you? Let me have a look here. Yeah. Here, look. Another thing I just wanted to show you. It's a little bit here. Boom. You know what this is? It is just regular sugar. Okay. And It's a little interesting. Look here, you see? Here it stays black. Yeah? It's kind of interesting, yeah? So, and where is my pipette? Here it is. Let's try the following. I need a little bit of water. So, and let's play a little bit. Let's see how this works. You know what? I'm going to put a little bit. I'm going to do it like this. I'm going to put some of the sugar on here. Ah, you see, if you put only very little sugar on here, then the exposure. Here we go. So, and look, I'm going to now add a little bit of water here on top. And then you're able to see how the crystals dissolve. And then when they start to recrystallize, but again, this takes a long time, then, yeah. But the sugar crystals also look nice. Yeah. And yeah, I think it looks looks kind of pretty as well, just playing around here. Yeah, yeah. So I'm reading again some of the thing. Vanta black, it's called. Yeah, I'll try to get some of this here. Yeah. So this kind of, you can see that the crystals start to dissolve, and then you can again see here that some of them display those nice, beautiful rainbow colors. Yeah. So. And then usually the dissolving stops when the liquid becomes sufficiently saturated. Yeah? It looks like ice melting, yes. Yeah, so what I'm going to do now quickly, did I check all of these things here? We need a dark paper, yes. What's the difference between front? Okay. Okay, let me have a look here. Yeah, so that's basically the thing here. Let me have a look now at the how do you call it, at the vitamin C again. Yeah, so yeah, you can play around with a lot of crystals, and what I also recommend is that you try to put a little bit of soy sauce under the microscope, like I have done some time ago, because the soy sauce contains a lot of salt as well and makes very beautiful crystals as well. Yeah. That's again the vitamin C. In the meantime, it has fully crystallized. So, yeah, another thing I wanted to show you very briefly. Again, this is an example of how depolarization works quite nicely, and that is, look at this here. These are feathers, but I need to put something over it. Where were my slides? Did I already use up all of them? Oh, here on the back they are. You just use a slide, you press it flat. And this is, again, I think one of a very nice example of how to increase the contrast. Yeah. So that's a bird's feather. Yeah. Yeah, so I just wanted to show you that yeah, you don't always need fancy polarizing substances to use Polarized light microscopy, but sometimes you can simply use it to increase the contrast like this. Cotton candy, many years ago. Just check the channel or search for it. I put some cotton candy under the microscope as well. I don't remember if I actually used polarized light or just dark field, but this was already several years ago. Just search for cotton candy, and one of the videos in this channel will be cotton candy under the microscope. Yeah, yeah. So that's yeah, bird's feather. I don't know which bird it is. Honestly, I found it somewhere, and simply, yeah, took it along. Yeah. feather mites, and not haven't found any mites. Yeah, yeah. So I saw a few dots. Are those? Honestly, I don't think so. That would rather surprise me. That, I mean, if you actually are able to see, see little animals with 8 legs, then these are definitely mites. So I cannot exclude this possibility, but that would rather surprise me, because this feather has been in my cupboard for many months. So this would have meant that they must have also survived for many months. Yeah. So generally, yeah, correct. All of these things here, those irregularly shaped structures, generally it's just some dust particles. Yeah. If you see mites, they look they look very clearly like mites. Yeah. Yeah. So, and those irregularly shaped structures that you see here, yeah, these are usually textile fibers or some dust. Yeah. Yeah. So this looks actually quite nice in contrast. Yeah. So I think the dog here I also showed you already before. Here it is. And again, I have to refocus again because this one is now in a small dish, and this changes the distance. Yeah. Nanoplastics. Oh, there are those nano and microplastics can be found, unfortunately, everywhere. Yeah, if you look hard enough. Yeah. But generally, yeah, there are so many dust fibers floating around in a household that they're almost unavoidable. And even if you start to clean your microscope slides, and sometimes it can be that, for example, if you use some tissue paper to clean your microscope slides, then you might actually find some of the cellulose fibers of this tissue paper on the slide. Yeah? So sometimes you end up contaminating the slide more. But if you know what you actually are seeing, then you can of course exclude these dust fibers as a possibility here. Yeah. Okay, what time is it? 1:07. Yeah. So bottom line is, I just wanted to. That's actually. I tell you a little bit of the background now. Some kind of a reason of why I'm actually doing now also some stereo microscopy is many people who are doing hobby microscopy, of course, they like to use compound microscopes. They're very popular. But stereo microscopes sometimes are a little bit, I wouldn't say ignored, but they do not seem to be quite as popular, right, as compound microscopes. And I simply like compound microscopes also because they allow you very easy and uncomplicated nature observation. So this is also one of the reasons why I occasionally do some sessions using compound microscopes, because I also would like to promote those. So for example, a couple of weeks ago, I was actually in, I made a video of this. I was for a full week, I was in Croatia for a marine biology week with a lot of students. And they had a whole range of microscopes there, and they were all stereo microscopes. So when we collect our plankton samples in little petri dishes, the students who have sometimes very little microscopy experience simply could take a dish with plankton samples and directly put it under the microscope. And the specimens were large enough to be seen properly, right? So it is, stereo microscopes are extremely practical, especially if you want to do microscopy with people who are not really experienced with microscopes, because you do not have to do any complicated sample preparation. You do not have to play around with the cover glass. You do not have to worry about the microscope objectives becoming damaged. Okay, so stereo microscopes actually, for promoting science, is actually have a very many advantages. Okay, just a second. I wonder if I find it here. Just a second. Just give me a second. I don't know where I put it. I just wanted to show you I misplaced it in my mess here. Just wanted to show you actually I think I might have it downstairs in the cellar. I wanted to show you actually one of the microscope objectives that was damaged by some students. Because they do not know how to use the microscopes properly, and if you do not instruct them well enough, then they end up damaging the microscope objectives. And this is something that can be almost completely avoided when using stereo microscopes, because the distance is quite large, and so there's almost nothing that can go wrong. So for here, it's better to use compound microscope. Yeah, it is. If you make for example, a slide, it does look better. The hair does look definitely better under compound microscope. There's no question. But again, you have to prepare the specimen, and for beginners, it might be an issue. Okay. Is it possible to use water microorganisms under the stereo microscope? Yes. Well, I mean, the large ones, I will tell you, I want to show you. You know what? I'll just not use I'll not use the thing here. I'm just going to use the regular, just a second, just want to show you how this looks like. This is how the water fleas look like with light coming from the bottom. Okay. So this is, and when you zoom in, then this is how they look like. So yes, of course it is possible, but it might not look quite as nice or as interesting as if you were to look at them using low magnification in a compound microscope. Okay, so this is just a on the other hand, the depth of field is also much larger. This means that it's easier for you to see the whole specimen in focus. So, you know what? Just for the fun of it, I'm going to turn off the light here now. Okay. And I'm going to attach now this ring light. And we're going to have... No, I don't even have to, because I have another one over here. I'll turn up the other light. This is now light coming from the top. Not from a ring light, but the built-in microscope light here. And you see this is how it looks like. So you see the contrast is not quite as high. So this is the reason why sometimes trying to increase the contrast somehow might be also quite something you want to try out. So here we go again. Again, the problem is a little bit the camera, which tries to adjust the exposure time all the time. So I'm reading again. So if you want to look at smaller water organisms, like for example, single-celled microorganisms like I don't know, paramecia or whatever, of course you are able to see them as well using a stereo microscope, but not quite as well, right? You just might see them as a small object. For example, there are some things floating around in between here. I don't know, if these are just some kind of debris that's kind of moving around with the water, or if they're actually indeed also single-celled organisms. I don't know. Okay. Have you ever heard about a mechanical stage for stereo microscopes? Yes, I've heard about that. This is also something where I also heard that some people actually either made one or bought one. Basically, a mechanical stage would allow you to turn something and then you're able to move it around. I say, why not? Try it out. However, I think that in most cases, it should be okay just to move it around with your fingers like this, because the magnification is not so high. But for a compound microscope, you almost definitely would need one for precise positioning. Generally, I found out at low magnification, it also kind of works to move it around with the finger. However, I understand for proper filming and so on, you see, it doesn't move quite as smoothly, right? So having some kind of a system where you can actually move it around nicely, smoothly would be kind of convenient. What I would like probably to have here is not just a mechanical stage where I can actually turn some knobs. This would not help me so much. But what I would like to have is actually maybe that's another project. It's a motorized control. The reason is I'm making those live streams, right? And I have to reach over to my microscope all the time. But it would be kind of nice if I have some kind of a joystick or so where I'm able to control the movement a little bit better, right? So I can essentially then look at the screen kind of and use some kind of a joystick. This would actually be indeed an advantage. Yeah. So I think you both have USB and industrial cameras. Do you notice any differences in quality? So what I have is the following. Yes, I do notice a difference in quality, but I do not think that this has to do something necessarily with the fact that it's USB or in an industrial HDMI camera. The camera that I have right now, of which I made a review in my other microscopy channel just a few days ago, I made a review. This is an HDMI camera, and I'm getting a significantly better image quality than with my USB camera, but not because it's HDMI or the other one is USB, but I think it has to do something a little bit with the age of the camera. There might be a couple of generations already in between. So this could be one reason. Another reason could be the following, and that is that many USB microscope cameras, USB 2.0 microscope cameras, essentially have a very high resolution. And this basically means that you can take nice pictures with it because of the high resolution. Let's say, I don't know, 5 megapixels or something. It's way more than you need. But this also means that if you want to make a video, you have to go down with the resolution again. And I sometimes found it difficult to actually get the proper frame rate because also USB 2.0 is a little bit slow. And so it depends really what you want to do. And generally for my purposes, I find that the current system works quite well. Okay, so I would say it's probably not a question of USB versus industrial HDMI camera, but maybe more a question of technology that's used, and also indeed the HDMI is quite fast. Yeah? But it depends really what you want to do. So I could not say that generally one is better than the other. I'm reading again here. I'm curious to see the effect of 1.5 volts electric current on the water fleas. Don't worry, I am planning on doing something like this because I do have a power supply which goes up all the way to 30 volts. So it's not only 1.5 volts, but maybe also 30 volts. And indeed, it is like this that Paramecia, for example, those single-celled microbes, they do respond to electrical fields. Right, they will move towards the cathode, which is the negative pole. So you can actually move them horizontally back and forth. And again, a few months ago, I think I did a live stream as well, where I experimented around with some of those Paramecia. But I could, of course, also try this with some water fleas and also with some nematodes. I've got some vinegar eels and some other microorganisms. But you know what's going to happen? Maybe as well, if you go up too much with the voltage, it might hurt the animals as well. So that's the thing. But electrotaxis, as it's called, is nothing unusual. So I wouldn't be surprised if they respond. But then again, if the voltage is too high, you might actually end up killing them because they do have a nervous system. I actually did once try that. I went a little bit too high with the voltage and resulted in the animals. Some of them, this was a flatworm. It stopped moving. It was a little too much. Yeah, they have a nervous system and therefore they do respond. Interestingly, Paramecium does not have a nervous system and still also responds. So this is But if you want to see the whole thing with electrotaxis in Paramecia, I don't know when I made that video. So. I blow the biological fuse, yes. Did you make a video about measuring the small dudes? Ah, that's a thing. You mean measuring like the size of them? It is possible to actually measure their size. I just want to show you something because you're asking. I hope I find it. Where is it? Here it is. If you want to measure something, then I recommend. What time is it? I'm always checking the time. That's the last thing I'm going to show you today. Because I do also. So this is a so-called a micrometer slide. And there is a small scale on it. And what you can do, of course, is you can. This is, of course, used for compound microscopes. But honestly, you can also use this for stereo microscopes, of course. Yeah. But over here, it's difficult to see maybe. It says here 1 division is 0.1 millimeter. Okay, so 1 division means, it's out of focus. Here we go. Yeah. So this is basically from one line to the next line. Okay, that's referred to as 1 division is 1/10 of a millimeter. Okay. Yeah, so basically the whole thing over here, from here to over here, is actually 1 centimeter. Huh? So this basically means that from here to over here, from 9 to 10, that's 1 millimeter. Okay? So, and with that, you know what, I'm going to just put this next to it or over it. So you see that those water fleas, so I just put the Petri dish with the water fleas directly over the micrometer slide. So you see that they are approximately 1 millimeter in size. A little bit larger maybe. Yeah. So, okay. So, yeah. I know I'm switching topics a little bit. I'm going away from polarized light microscopy. It doesn't matter, right? So you see, I don't know, 1.5 millimeters maybe. Yeah. So, and very interesting, am I the only one to feel guilty to kill microorganisms? I think that's an important question, and maybe right now at the end I'm going to talk a little bit about that, because this is indeed a question. You see, I'm a biology teacher, right? And when we look at microorganisms in class, or even catch insects, and we have to kill them, of course some students don't like doing that. And they have certain concerns about ethical concerns, and I think it's generally good to have ethical concerns, animal well-being and so on. However, I do make a difference between true microorganisms and microscopic and small animals. Real microbes like Paramecia and so on, they do not have a nervous system, right? And basically, they do not have an individuality as we know it, right? No personality and so on. And I do not feel guilty about killing those because, if you put them back into water, fish eat them, they're going to die anyway, because you should not or cannot see microorganisms as, how shall I put this into easy words, as individuals. You see, when I scratch myself, that's the example I'm giving, okay? I'm scratching myself. I'm killing thousands of skin cells, and I'm not feeling guilty about those either, right? Because this concept of individuality does not exist on that level. And that's what I'm kind of saying is I think while it is good to think about the ethical issues about animal welfare, and microorganisms are not animals strictly speaking, but you get my point, is while I think it's important to kind of consider this as well, I think it's also important to worry about the real issues, and the real issues are, I would say, the higher animals. I mean, because of pollution and I don't know what, so many, we really have a lot of biodiversity dying, Okay, and many microbes are not endangered. Okay, so what I'm saying is let's feel guilty about those organisms that actually are really endangered. Okay, that's kind of my view about this. While at the same time, I do understand that, of course, once you look at those water fleas, I mean, I'm not going to flush them down the toilet now. Yeah, I'm going to put them back into the jar. Yeah, obviously, but at the same time, I also know that by natural progression, many of them are going to die because that's simply the normal thing that happens, right? So just a couple of thoughts that I have here. And when I eat yogurt, I eat bacteria, and in the stomach, acid kills billions of them, right? But not a problem, I say, because they do not have an individuality, because they do not have a nervous system. Yeah, these are just bacteria under quotation marks, right? So I think what we have to do is we have to kind of differentiate a little bit which type of microscopic specimens we're looking at. But then again, of course, also microbial biodiversity is worthy of protection here. Yeah. So that's the thing that I just wanted to Thank you, Oliver. I was shy to ask this question, and I love your answer. Thank you. I'm happy I was able to help you. I can just tell you this is not this question about ethics and microorganisms and microanimals is not uncommon, right? I will tell you the following. We actually, even in a few days, we're going to do an insect project in school where we're going to catch insects with the help of an insect specialist who will decide then which insects are worthy of protection and which not. And we're going to put them under the microscope. And in order to do that, we have to unfortunately kill them. But we're not going to kill all of them. We're only going to kill those that actually are going to be not protected. That's why we need an expert to help us here, because we want to stay ethical. But at the same time, I have to say the following, and that's why I think it's, for me as a biology teacher, is so important. This is if people, students and so on, are not exposed to insects, to microorganisms and so on, then they will also not value them. Yeah? And then for many people, microbes, water fleas, microorganisms are, they don't like that. They feel disgusted or whatever. And I think if we're able to just view them as what they are, beautiful organisms, then I think, because we've been observing them, then I think that's also something that's important. Yeah. Okay. I like to ramble. Okay. Very quick. Tardigrades. Yes. I'm going to collect some more. Yeah. Mollies, I used to have mollies when I had an aquarium. Yes, they used to eat their own babies. Okay. So just saying, I'm going to quit now. 1.5 hours goes by very quickly. Organizational issues again. Next week, I do intend to do it again on a Saturday. Okay. Please do check the community posts and the announcements. Occasionally, I have to move it to a Sunday. But I hope I can go back again to the regular schedule. I'm going to call it quits for today. I wish you all the best. Happy microbe hunting as always. Enjoy microscopy. And hope to see you again around. Okay? Bye-bye. All the best.